everyone. I hope you all are doing great. I am Kavya Gupta from yICSA, and I'll be your host for the day. We are extremely happy to resume our webinar series and looking forward to many more amazing talks from all of you on our platform. If you're also interested in sharing your latest research on our webinar series, then you can mail us at yicsa.committee@gmail.com. So before I dive into introducing our speakers of the day, I would like to request and highly encourage all of you to use the option of raise hands after the talk gets over in order to ask questions. So for today, we have Gabriela Desdin-Mico with us. She is currently pursuing her PhD in Maria Mittelbrun's lab from University Hospital, October 12. And the title of her talk is Immunometabolic T cell failure induces multimorbidity and aging. So Gabriela, welcome. And you can share your screen now.
Hi.
Hi.
So I can start?
Yes, Gabriela.
Okay, good afternoon. My name is Gabriela Desdin-Mico, and I am presenting this webinar titled Immunometabolic T cell failure, induced multimorbidity and aging. I'm from the Immunometabolism and Inflammation Lab that is led by Dr. Maria Mittelbrun, and we are interested in inflammation. So inflammation, when it's acute or is local, has beneficial effects because allows you to fight against pathogens and also allows you to solve some injuries that need to be resolved. But also have been linked that when you have chronic inflammation, that have been linked with several pathologies like cardiovascular diseases, neurodegenerative disorders, or autoimmune diseases, and others. All these pathologies has three features in common. One of them is that immune cells are involved. The second one is that tissue homeostasis is lost. And the third one is that these pathologies, the incidence, tend to increase as we age. That is why we know it as age-associated diseases. So I guess that everyone knows that when we age, we tend to increase the level of pro-inflammatory cytokines in our serum. This is a low but chronic inflammation, and we know it as inflammaging. As you know, aging is associated with chronic inflammation. And also there are other studies that links anti-inflammatory cytokines like IL-10 to longevity. So we are interested in the role of, we want to know if inflammation is a biomarker or is a direct driver of aging. And also we want to know what is the contribution of immune cells to aging. Mainly we are interested in T cells among other immune cells. Immune cells have the particularity that they can travel around all the organism. And once they found a threat, they can activate it, proliferate it, and then they can differentiate into different subsets. So T cells tend to differentiate it to different types, like Th1, that this is a highly pro-inflammatory T cell subtype with secreting higher amounts of pro-inflammatory cytokines like TNF-alpha and interferon gamma. So recently, the immunometabolism field have take interest because we know that manipulating the metabolism, we can control the activation of immune cells. For example, now we know that T cells, when they are in a naive state, they use mainly oxidative phosphorylation in the mitochondria, and they are naive, they are not activated. Once they need to be they activated for a threat, they tend to proliferate and activate it, and they change the metabolism. They become highly pro-inflammatory cells. And then once the threat is finished, they change again the metabolism, and they become again, well, they start to use oxidative phosphorylation in the mitochondria. That is a normal situation, and in that situation, you are an immunocompetent individual. However, as you might know, when we age, we also tend to have mitochondrial dysfunction. So if you have a T cell that is naive, that is not being activated, but the mitochondria is dysfunctional, maybe when you activate it, then you cannot come to again to a resting state, and you maintain being a effector. So there is a lot of new papers that links that glycolytic immune cells are highly pro-inflammatory. So our hypothesis is that when we age, T cells, they have mitochondrial dysfunction, and they become really glycolytic and pro-inflammatory. So there is a lot to know about mitochondrial dysfunction in other type of cells, but in T cells, until recently, it wasn't described anything. So we are interested in mitochondrial dysfunction in T cells. So first of all, we analyze T cells from age wild types around 22 months old. So we look at the mitochondria, and we measure mitochondrial RNA, that these two subunits are encoded in the mitochondrial DNA. We measure in T cells, and we compare with controls. And what we observe is a decline. We also perform seahorse analysis that allow us to know if these T cells use mitochondrial metabolism or not. What we observe is that they don't use it. So we also wanted to know what happened when all your T cells are dysfunctional. For that, we generated a mouse model where all T cells are dysfunctional. And for that, we took advantage of the transcription factor, that is TFAM. This is a transcription factor that is encoded in the nucleus, but goes to the mitochondria. And this transcription factor is the one that is on charge of the mitochondrial DNA transcription, replication, and stabilization. So if you don't have this transcription factor, you will have a mitochondrial dysfunction. So T cells from these animals have low levels at mRNA levels of TFAM. So we also observe that in normal age T cells also have mitochondrial dysfunction with the metabolism is glycolytic, and they don't have oxidative phosphorylation metabolism. And we also observe that these T cells become really pro-inflammatory in the type of Th1. So this is data from T cells coming from aged animals and our young TFAN CD4 KO mice. We haven't do any challenge, and what we observe in a steady state is that these T cells are more Th1 expressing Tibet and interferon gamma and TNF alpha. That is the same that happened in aged T cells. So next, we wonder in vivo what happened with these animals. These animals are able to if we do it immune challenge, how they will respond. So we challenge the animals with this virus that is ectromelia virus. It's a highly virulent mouse pox virus that causes a disease similar to human smallpox. So when we challenge, we use young wild types, all wild types, and our young T-fan CD4-KO mice. We infected in the food pot, and what we observed is that these animals died, all wild types and our young T-fan CD4-KO mice all died between day 9 and 10. And also they don't, in the food pot, they don't have a local inflammation. So these animals are immunodeficient. Also, we measure in the serum of these animals, but without any challenge in a steady state, the levels of pro-inflammatory cytokines like IL-6 and TNF-alpha, and we find an increased level of these cytokines. So we wonder what happens if you have an aged animal with mitochondrial dysfunction. So we let these animals to get old, these transgenic mice, and we monitorize them. So we observe at 7 months that these CD4 T-fan KO mice were fragile, they have age appearance. Also, we observe that they presented kyphosis, also anemia, they weight loss, also females and males. Also, we make some metabolic studies, and we observe that these animals tend to have less activity, and also when they move, they move slower than controls, and tend to live less than controls. So with all these data, we propose that these animals suffer from a premature aging, and it could be this premature aging could be due to this chronic and low inflammation that they have, that tend to increase as they age. So as you might know, inflammaging is a risk factor for multimorbidity. There are some studies that links that if you have higher level of pro-inflammatory cytokines, you will have multiple comorbidities, and it's also a hallmark of it is indicative of frailty and premature death. So we wonder if in our mice we have the same phenotype with some type of multimorbidity. So we start the study. The first, we analyze the muscle, and we observe that these mice have a slower fiber area. When we analyze the glucose metabolism in vivo, we observe that tend to uptake less glucose compared with control. Also, they have less grip strength, and they tend to, this muscle is highly catabolic. So we have muscle wasting. Other tissue that is really affected in these mice is the adipose tissue. So we check in vivo by magnetic resonance imaging the percentage of adipose tissue, and we observe a reduction of this tissue. Also in the histology, we observe a reduction of the adipocyte site area. So we wonder what happened with this adipose tissue that these animals are losing. So we check for lipolysis. We check for ATGL, that is the first enzyme in the lipolysis pathway, and we observe that in these animals is upregulated. So we also corroborate the signs of lipolysis with measuring in the plasma of these mice the free fatty acid, and we observe that is increased. Also, these animals have heart problems. The heart is smaller and tend to pump more. The heart rate is faster than the controls. Also present aortic dilatation that is related with a reduce in blood pressure, fibrosis in the aorta, also decreased contractile markers, and we also observe partial dissection in the tunica media of the aorta with higher foci of inflammation. Then we wanted to know what happened with this animal, why with the mitochondrial dysfunction exclusively in T cells, why these mice age prematurely. So we perform our RNA-seq that I'm not showing, but in there we observe an upregulation of genes related with senescence. So we measure senescence markers like P21 in several tissues, and we observe an upregulation of these markers. We also check for beta-galactosidase, senescence-associated beta-galactosidase activity, and we observe that is increased in white adipose tissue and in the kidney of these mice. So we know it that these mice have this multimorbidity phenotype, but we don't know why. So we tried to link. So we found this paper that is from Raumuller. So in this paper, they describe that Th1 cytokines, interferon gamma, TNF alpha, that are secreting that Th1 T cells secrete. These cytokines are able to induce senescence to cells that have been suffered from some malignant transformation. So we wonder if that could be in this model the mechanism. To address these questions, what we did was we cultured different cell lines with serum from control animals and our T-fan CD4-KO mice, and we observed an increase of P21 in these lines. We also observed, we also did the same experiment, but we had an extra condition where in control serum we added 2 TNF alpha, and we observed that the levels of P21 were increased. Then we tried to check if this phenotype was only due to immune cells. We used several strategies. One of them was bone marrow transplant. For that, we transplant bone marrow from control mice and also use bone marrow from T-fan CD4-KO mice. And we transplanted 2 T-cell deficient recipients. And these mice don't have any T cells. So we check after reconstitution. So without reconstitution, we don't have CD4 and CD8 T cells. And we have it in these 2 situations. And we let these animals to age. And we observe that we recapitulate all the phenotype that we see it in our mice. We observe an increase in senescent markers in several tissues. We also observe a reduction in body weight and grip strength in these animals. And we also find increased levels of pro-inflammatory cytokines in the serum of these animals. So we wanted to revert this phenotype, so we checked for some therapies. As you might know, that when we age, that pro-inflammatory cytokine increase, but there is also a dysfunction in mitochondrial function, and treatment of progeroid mice and also different age-associated diseases with NADA precursors. This treatment tend to revert the phenotype. So we decided to check if we have a reduction in the NAD levels or in the ratio NAD-NADH. So we measure in the liver of these animals, and we observe that is decreased also in T cells. So we decided to treat these animals during 10 weeks with one of the NAD precursors, that is nicotinamide riboside. So we increase the ratio in the liver, a little bit in T cell, but we don't rescue T cell phenotypes. However, we revert all the cardiovascular defects, the activity, the metabolic parameters, and also the strength of these animals with this treatment. So what happened with senescence? So we observed that in these animals, when we treated with this NAD precursor, we have a reduction in senescent markers, and also a reduction in the serum levels of pro-inflammatory cytokines like TNF-alpha. So even though we cannot revert the phenotype of the T cells, we believe that these T cells induce senescence, and these cells that become senescence become highly pro-inflammatory. So if we treat it with this NR, so we don't allow that these cells become senescence. So that is why we observe a reduction in circulating levels of pro-inflammatory cytokines and also markers of inflammation in the liver of these animals. So we propose that this new model could be used as a new model of aging and senescence, and we can use drugs that allow us to manipulate the metabolism of T cells to reduce inflammation or try to prevent age-associated diseases. So thank you very much for your attention. I want to thank to my mentor, Maria Mittelbrum, for allowing me to do my thesis with her. And also I want to thank to my all my co-workers, because without them we couldn't do anything. So thank you very much.
Thank you, Gabriela, for the amazing talk. So we have a question from Staffan Stromblad. I hope I pronounced it right. You can unmute yourself now.
Wait a moment.
Thank you. So I'm Staffan Stromblad from Karolinska, Stockholm. Thank you very much for this presenting its exciting study, very elegant. I was wondering about the potential treatment that you used. Do you think that would be any adverse effects by increasing NAD in short or long term if we would use that as a prevention?
To induce, sorry?
The treatment you made to increase the NAD levels?
What? I don't understand.
If do you think such a treatment, if we would try to use that as a preventive measure, may have any adverse effects in short or long term?
I think that maybe cancer cells would proliferate, but I think that maybe we need more trials, more studies. I can say that we also treat wild-type animals with this drug, and we observe a reduction in pro-inflammatory cytokines, and during the necropsy we didn't observe any tumor, but I'm not 100% sure. I don't know if I answer correctly your question.
Yeah, of course you couldn't know. But yeah.
I only can answer by our studies.
Yeah. Oh, thank you.
You're welcome. Thank you, Stefan. We have another question from Marta.
Hello. Can you hear me now?
Yes, Marta.
Okay, great. Hi, thanks so much for that lovely talk. I actually had two questions to ask you. So one was the pro-inflammatory cytokines that you're seeing at steady state levels in the serum of these mice. So do you think that these are actually coming from the cytokine storm of the T cells, or they're coming from the senescent cells that are being produced as a consequence of the cytokine storms?
I think that they might come from T cells, but also T cell activate other immune cells, and also from senescent cells. I think that maybe came from these three different places.
Because when you guys do the NAD treatment, you see a reversion of the circulating cytokines, but you don't see a suppression of the pro-inflammatory phenotype of the T cells. Is that correct?
Yeah, yes, that's correct. That's why we say that maybe we are preventing senescence. So if you prevent that cells become senescence, you don't have a higher amounts of pro-inflammatory cytokines. That
Okay,
could be another possible explanation. Could be that also this treatment tripped off, other immune cells that in that moment are pro-inflammatory due to the Th1 cytokine storm.
Okay, but so what it's suggesting then is that the systemic inflammation basically that we can measure in the serum is coming from other cells, not the T cells themselves.
Not exclusively from T cells. That's the point.
Okay. And my other question is, you showed nicely that there was some accumulation of senescent cells in the organs. Did you check in vivo senescence after the NAD treatment?
In vivo after, but in our transgenic mice model, or in the
in the transgenic mouse model.
But in vivo, do you mean like
in the organs? For example, I think you showed, what was it, a liver maybe that was quite beta-gal positive?
No, we check in the liver for P21.
Okay, and that was reduced after the treatment.
Yeah, it was reduced, and P53 also was reduced.
Okay, thank you so much.
You're welcome.
Yes, Gabriela, we have another question in chat box from Izumi Horikawa. She is asking, what cell types mainly produce or secrete IL-6 and TNF-alpha, dysfunctional or aged T cell, or other cell types like macrophages?
I think that TNF-alpha, it could be secreted for a lot of cells, like adipocytes, preadipocytes, macrophages. So it's like a lot of cells can secrete these cytokines.
I hope that answered her question.
Now
we have another question from Corina. Really exciting work. Do you think T cells, T-FAMFL, themselves have also become senescent? And what's the effect of the T cells on other immune cells, like macrophages, for example?
We have not checked if other immune cells are activated and what are the effects. We are starting to see it. And also what we observe is that these animals have pro-inflammatory foxes, and we mark for CD3 cells, and we find it, but we find another cells that it could be macrophages, but we haven't looked so close.
Perfect. Thank you so much, Gabriela, for being with us today. We are extremely elated to have you here. Yeah, thank you. So guys, we'll move on to our next talk of the session.